Okay, I'll do a quick introduction here. Great pleasure to have Professor David Allsop from Lancaster University do a presentation. As I've already stated in my presentation, I think it's very important to actually see why we're actually going and doing all this measurement. It's all very good measuring nanomaterials, nanoparticles in the environment, but why actually would you want to? I gave a bit of an overview that there's potential around particles to have potentially different levels of toxicology, and they may get into parts of the body that other particles and substances won't. Well, here hopefully we have a potential actual real-life situation. So quickly introduce Professor Allsop, who's been working around Alzheimer's for nearly 40 years. He was the first person to actually isolate the amyloid plaques in the human brain that have been probably beyond strongly linked. I think it's now a definitive actual symptom of Alzheimer's, although he may well correct me during his presentation, in the 1980s. And a particular interest at the moment, among many other things around Alzheimer's, is the potential of redox active metal ions to have a link with Alzheimer's, and therefore that also links in with nanomaterials, especially magnetite, magnetic nanomaterials, because they do display quite a strong redox potential. Quick overview. So as you saw, 1980s, doing postdoc at Nottingham, which is where I presume you did the isolation of the amyloid plaques. Then done quite an extensive academic trip around the world by the looks of it. So it's been in San Diego, Japan, Northern Ireland, a brief spell in industry by the looks of it at Smith Pine Beecham. Then has been at Lancaster University since 1998 in the Department of Biological Sciences. So presenting pollution-derived magnetite nanoparticles as a possible risk factor for Alzheimer's disease. Thank you very much.
Okay, thank you for inviting me, and thank you for your introduction, Neil. So I'm a biologist, I'm a biochemist, and I have a long-standing interest in Alzheimer's disease and what goes on in the brain in Alzheimer's disease. And this potential link with magnetite nanoparticles is something fairly recent due to a collaboration with Barbara Mayer here. So if I can have the next slide. This is the main focus of my research. So it's on particular proteins that accumulate in the brain, not only in Alzheimer's disease, but in other diseases as well, like Parkinson's disease and CJD, which is the human equivalent of mad cow disease, Huntington's disease, and so on. And in all of these different diseases, you get a particular protein, or sometimes more than one protein, accumulating in the brain. So these proteins change shape, they become misfolded, and they start to aggregate together to form these fibrils, fibers that accumulate in the brain, often referred to as amyloid fibers. But in each disease, you get a different protein accumulating. In Alzheimer's disease, you get more than one protein, so you get these things called plaques, senile plaques in the brain, and you get these things called neurofibrillary tangles in the brain. The plaques are found outside nerve cells, the tangles are found inside nerve cells. The plaques are made out of this substance called beta amyloid. The tangles are made out of a different protein called tau. And we think these things are very important in the pathogenesis of the disease, because mutations in the gene for each of these proteins give rise to inherited forms of disease. And there's no doubt that these proteins are actually the cause of prion disease. So things like Creutzfeldt-Jakob disease in humans, the actual infectious agent that causes those diseases is this prion protein, which is one of these misfolded proteins. So if I can have the next slide. So one of my areas of interest since I moved to Lancaster has been the role of metals and metal ions in neurodegenerative disease. And there's evidence for extensive oxidative damage to the brain in a range of these different diseases. And of course, redox active metals from generating reactive oxygen species and causing oxidative damage. And the brain is particularly vulnerable to this type of damage because it uses large amounts of oxygen and has relatively low levels of protection by antioxidants. And the interesting thing is that the plaques and the tangles that I've mentioned in Alzheimer's disease are sites of redox active metal ion accumulation. So you can find things like iron at the site of the plaques and the tangles, and also many of the key proteins that I mentioned are metal binding proteins. So they have high affinity for certain metal ions, and that includes the proteins that you find in Alzheimer's disease, in Parkinson's disease, and so on. So there's quite a well-established connection between metal ions and these diseases. And this area of research in my lab was really started off when a PhD student of mine came and showed me this paper. So he was a chemist, and he came and showed me this paper from Ashley Bush and colleagues, who claimed that the beta amyloid protein, peptide, that accumulates in the plaques in Alzheimer's disease can produce hydrogen peroxide through metal ion reduction. So this protein can interact with redox active iron or redox active copper ions, and then can generate hydrogen peroxide from molecular oxygen through these sort of redox mechanisms. And people were very skeptical about this at the time, but through a collaboration with, go to the next slide, through a collaboration with a guy in chemistry called Brian Tabner, we set out to determine ourselves whether these amyloid proteins could generate hydrogen peroxide. And the technique we used was electron spin resonance, ESR, or EPR, it's sometimes called spectroscopy. And this involves incubating this peptide for various periods of time to allow it to generate any hydrogen peroxide, and then stimulating this reaction here, the Fenton reaction, by adding iron, and then using a spin trap to detect any hydroxyl radicals generated. And then on the next slide, you can see the characteristic spectrum that is generated in the presence of hydrogen peroxide. So this is simply the Fenton reaction here. And when you carry out the same reaction with the beta amyloid, you generate the same spectrum, the same 4-line spectrum, which shows unequivocally that this beta amyloid protein does actually generate hydrogen peroxide and hydroxyl radicals. And we did various controls involving adding catalase to destroy hydrogen peroxide, adding metal ion chelators, and so on, and showed quite convincingly that these things do generate hydrogen peroxide and hydroxyl radicals. And just to cut a long story short, we have a whole series of papers on different proteins and different diseases. So this applies not only to Alzheimer's disease, it also applies to the proteins that accumulate in Parkinson's disease, prion disease, which is the CJD in humans, and various other things, even type 2 diabetes. You get one of these proteins accumulating in the pancreas in type 2 diabetes. So all of these things can interact with metal ions, and they can all generate reactive oxygen species. And that, of course, may be one of the mechanisms for damage to the brain or other tissues in these diseases. So this is where the magnetite starts to come in to the story. So Barbara knew that I worked on this type of thing, and she came to talk to me about magnetite in the human brain, which is, of course, a form of iron, iron 2, iron 3, mixed iron oxide, and was first detected in the human brain in 1992 in the form of these characteristic shaped, euhedral shaped, sort of cuboid shaped crystals here, and it was thought that they may play a role in iron storage. Since then, it's been shown that they may play a role in navigation in birds, so detecting the Earth's magnetic field. They may even play, there's been suggestions that it may even play a role in memory processors. So this magnetite has been shown to be present in the brain for some time, and this was thought to be generated through purely biological processes. So there's no suggestion that this would come from an external sort of pollution source. And if I can have the next slide. And then, this is why Barbara came to talk to me. Papers started to appear in the literature looking at magnetite in brain tissue from cases of Alzheimer's disease. So this is using this squid magnetometry technique. And here we have 3 cases of Alzheimer's and 3 controls, so we only have very, very small numbers here, so it's difficult to give any definitive link with the disease. But in these 3 cases of Alzheimer's, particularly this one case here, there seem to be elevated levels of magnetite in the brain. But again, there was no suggestion here that this was anything other than normal biogenic magnetite. If I can have the next one. That's it. And then papers started to appear from a group in Mexico City suggesting that living in a highly polluted environment, city environment, might be linked to changes in the brain that are indicative of early stages of Alzheimer's disease. So residents of cities with severe air pollution, for example, Mexico City, you can see increased levels, for example, of this beta amyloid protein starting to accumulate in the brain. So this isn't fully advanced Alzheimer's disease, but it's suggestive of a very early stage of Alzheimer's disease. And you can see here, this is a low pollution environment, and this is a high pollution environment where you get much more of this amyloid protein starting to accumulate. And if I can have the next slide. So we decided to investigate this ourselves a little bit further. So this is Barbara and myself, and other researchers were involved as well. For example, in Manchester, where we obtained some brain tissue from. And I'm going to show you some of the data published in our paper, our PNAS paper in 2016. So this is samples of brain. Some were obtained from Mexico City. So these are quite young individuals. They don't have diagnosed Alzheimer's disease, but they're young individuals that have lived in this highly polluted environment. And there are other cases from Manchester that do include clinically diagnosed Alzheimer's disease or other conditions which have some aspect of Alzheimer pathology. And we measured the brain magnetic content using this squid magnetometry, which Barbara uses. And we also extracted the magnetic brain particles from these brains using a magnetic extraction technique. And we looked at those by electron microscopy and various spectroscopy techniques. And each analytical step was, of course, designed to minimize magnetic contamination from the environment, but of course it's very difficult to exclude that completely. Okay, if I can have the next slide. These are the squid measurements. There's no attempt here to link this to Alzheimer's disease or Alzheimer pathology. So we don't have a range of control brains and a range of Alzheimer brains. The take-home message here is that it's easy to measure magnetite concentration in the brain using this squid technique. Some individuals have high levels. So this is a young resident of Mexico City. Some have relatively low levels. High levels could be detected in some Alzheimer brains. High levels could be detected in some of the younger residents of Mexico City. If I can have the next slide. And then we started to look at microscope images of these brain sections. And this is a collaboration with Glasgow University. And we saw this magnetite here, which is similar to previous reports of biogenic magnetite. But we also saw lots of these other spherical particles, which were very different in appearance to this biogenic magnetite. So these are small spherical particles which don't seem to have been previously described in human brain. These clearly are magnetite, so you could do the spectroscopy on them, and you can demonstrate that they are composed of magnetite, both these biogenic magnetite and these small spherical particles. They're both a form of magnetite. If I can have the next slide. And the extracts that we made from brain tissue, so these are extracted magnetically, so you make an extract of the brain cortex and then you pass it over a magnet and you extract these things from the brain. We saw large numbers of these small circular magnetite particles, and you can show quite clearly that they are magnetite, and not only the spectroscopy but also the lattice arrangement here is consistent with these things being magnetite. The size distribution is here, so it's quite broad, but a median longest diameter of around 18 nanometers. And this kind of dimension greatly exceeds those of iron particles formed within the ferritin, which is an iron storage protein. So they can't really be produced by biological processes. And if you look at the next slide, the surface appearance of these things, and this is, I'm going by what Barbara tells me, so it's her area of expertise rather than mine, but the surface appearance of these things is identical to what she has seen from magnetite extracted from the air and from the environment. So they have a very distinctive surface texture, and these have not been identified previously in human brain sections. They're difficult to reconcile with natural biological processes, and they bear a compelling resemblance to magnetite nanospheres found in airborne pollution. And so if we look at the next slide, these are magnetite particles that Barbara has actually extracted from the environment. And you can see this image, this scanning EM image here. And if we go back to the previous slide, this is compellingly very similar to this image here. And so it's highly suggestive of these things having an external source rather than being produced naturally within the brain, although that isn't, of course, proved definitively. So if I can have the next slide. And when we first submitted this paper to PNAS to get it published, they said, well, why don't you look for other more exotic metals in there, other than iron, and if you can show that those are present, then it would be more convincing that they come from a pollution-derived source. And so we had a look at that, and we found other particles which are not iron related, for example, derived from platinum, nickel, and cobalt, and there's no real mechanism for regenerating those things biologically. And this was even more suggestive that they might have an external origin. If I can have the next slide. So how do these things get into the brain? Well, it's quite easy to conceive how they might get into the brain. So if you breathe them in through your nose, they will enter the olfactory areas of the brain. I mean, this is a well-developed drug delivery mechanism. If you want to deliver drugs to the brain, you can sniff them straight up your nose, and they go into your brain. They can be transported throughout the brain, so they can move once they're in the brain, and they can potentially accumulate in regions of the brain that have been connected with diseases like Alzheimer's disease. So with this delivery mechanism, there is no blood-brain barrier preventing the passage of these things into the brain, so that they potentially have a direct access to the brain. And if I can have the next slide. During the preparation of our manuscript, another paper appeared by this group here, who found similar objects to the ones that I've just described to you, actually accumulating within the amyloid plaques that are found in Alzheimer's disease brain. So this is what these amyloid plaques look like. These tiny little dots here are made of magnetite, so that's shown definitively by spectroscopy, and they actually accumulate within the amyloid plaques, which are causative of Alzheimer's. Now, this could just be coincidental, because in Alzheimer's disease you get huge numbers of these plaques in the brain. You also see quite large numbers of these small magnetite particles. So this finding could just be through random association. So that there's no real attempt here to show statistically that there's a real association with the amyloid plaques. But it's quite an interesting and suggestive sort of observation. And if we can have the next slide, there's also quite a lot of support from epidemiological studies of a link between exposure to air pollution and dementia. I think you may have mentioned that. I mean, there's quite a nice study here published in the Lancet by a group in Ontario, Canada, where they've looked at incidence of dementia and they've looked at distance from a major traffic road. So this is meters away from a major traffic road, and this is the hazard ratio for dementia. And so you can see you have an increased risk of developing dementia if you live 50 meters or so away from a major traffic road. And this risk diminishes the further away you go. And by the time you get to 200 meters or more, then you have no increased risk of developing dementia. And this is really quite a nice study. I mean, it was published in the Lancet, so it's a very well controlled study. This association was only found with dementia, so it wasn't found with Parkinson's disease or other brain diseases like multiple sclerosis. Of course, this doesn't tell you what aspect of the so-called traffic pollution or whatever is linked to dementia. So it could be noise pollution, it could be some other environmental pollutant, but I mean, this is quite interesting and suggestive as well. Okay, and next, I think we're getting towards the end. So, these redox active metals play quite an important role in Alzheimer's and other brain diseases. The plaques and the tangles that you get in Alzheimer's are sites of accumulation of these redox active metals, and many of the key proteins involved in these diseases are actually high affinity metal binding proteins. So the prion protein that you get in prion disease, CJD, is the highest affinity copper binding protein known. Very, very high affinity metal binding proteins. These can, of course, generate reactive oxygen species through metal ion, changes in metal ion activity, and more recently we've found these magnetite nanoparticles in the human brain, which are remarkably similar to those present in airborne pollution. Living close to a major road increases risk of dementia. So the obvious question is, could these be a risk factor for dementia and diseases like Alzheimer's disease? I mean, the link with magnetite in particular is quite tenuous at the moment, but there is increasing evidence that traffic pollution in general is linked to dementia. And I think I'll finish there.
Thank you.
Thank you.
What are the next steps for this? If we're talking about traffic pollution, as electrification of the fleet happens, is that going to change the way that these risk factors need to be calculated in the future and forecasted?
Well, what we're doing, we have a PhD student at the moment that's, first of all, we're looking to see if there genuinely is a link with Alzheimer's disease. So we're now looking in brain tissue from Alzheimer cases and also controls, and we're looking at association with presence of plaques and tangles and that sort of thing. So those are being measured by Barbara using her squeak magnetometry with brain tissue from the pathology unit in Manchester. I think your other question, Barbara, would be better placed to answer that. But undoubtedly, the electrification of vehicles will presumably reduce, hopefully reduce this potential risk.
Just a quick question before we move on. Firstly, I have to say that everyone going to the conference last week, reactive oxygen substances and oxidative stress are actually linked. It's one of the main mechanisms regarding quite a lot of nanomaterial and particulate toxicity beyond Alzheimer's. A lot of inflammation and inflammation that can then lead on to cancers is related to ROS, and also ecotoxicity is also related, as this is as a mechanism. So my actual question is we deal quite a lot with sort of endpoint toxicology studies to try and identify whether a substance is potentially toxic or not. So this touches on carcinogenicity, mutagenicity, something. Well, are there in research any in vitro studies to actually try and link between an exposure to a chemical and Alzheimer's? Do you know if there's anything out there that's being researched that might in 10 years or 20 years time become an OECD tester to test all your chemicals against?
Not so much Alzheimer's, but I mean, there are better links, better established links with things like Parkinson's disease. So in Parkinson's, there's a well-known potential link with herbicides, pesticides, and that are actually causative of Parkinson's disease. Not so much with Alzheimer's.
Can you get a cell line that is perhaps made particularly sensitive?
That's one of the other things that we're doing. So we're doing cell cultures, and Barbara is extracting these things from Lancaster city centre, and we're aiming to apply them onto cultured cells and see what type of damage they produce. But it won't be surprising at all if they produce some sort of oxidative damage. I mean, that would be highly anticipated.
So I was saying about Carlos's question in my presentation about potential future directions. REACH has a lot of leeway for emerging hazard areas. So there's always a reference to equivalent level of concern. So say in 25 years time or 20 years time, the technology's got and research has got far enough for the OECD to be happy to produce a really well validated test to go and test for potential of forming of Alzheimer's or Parkinson's, then that is the way that the regulations may well suddenly go and say, oh, actually, we've now got a really good study to go look at another health impact that we're concerned about, and that's one area that the regulations might be adjusted. Thank you very much. Is that everyone? I know that Douglas is wanting to head off relative to London. Are you still okay? Yeah, okay. Yeah. Brilliant.
Sorry, I have to disappear now because I've got another meeting to go to as well. But around, if anybody wants to contact me for any reason, I'm around at the university.
Yes, well, we got you. Thank you very much for your time. Thank you.